Welcome everyone. My name is Associate Professor Jeff Craig and I lead the Early Life Epigenetics Group within the Impact Institute at the School of Medicine at Deakin University. I also lead the Gene Equality Deakin University Research Network. My Gene Equality colleague, Associate Professor Neera Bhatia, and I are organizers of this Healthy Futures Seminar series. So welcome to the series. I'd like to begin by acknowledging and paying my respects to the traditional custodians of the land on which each of us are today. I would like to pay my respects to the elders past, present, and emerging. Let us pause to learn, acknowledge, and celebrate their ancient histories and cultures. For those that have not attended one of our seminars to date, a quick rundown on the structure of today's event. In a moment, I will introduce Deputy Vice Chancellor of Research at Deakin University, Julie Owens, who will then introduce our speaker, Merlin Crossley. Merlin will present for 30 minutes. We'll move to some discussion with our panel and then to questions and answers. So I'll be chairing the discussion on Zoom and my colleague Tao Fan will be moderating the live YouTube chat. We're very keen to answer your questions. So you can either post your questions on the YouTube chat, send them to us on Twitter at SSN Deakin. Make sure to use the hashtags, hashtag SSN Seminar and hashtag Healthy Futures, or email to ssn-info@deakin.edu.au. I'd now like to introduce Deputy Vice Chancellor for Research and Alfred Deakin Professor Julie Owens.
Thanks very much, Jeff. Today, the world faces one of the most disruptive challenges globally that we've had for many decades, the COVID-19 pandemic. It's a global challenge, yet one that impacts everywhere in the microcosm, locally, affecting all of us directly and indirectly. Now, key to addressing the immediate and longer term impacts has and will be the role and contribution of researchers, of social researchers, as well as scientists, also economists and political scientists, as we try to mitigate the public health impacts, but also those wider economic and social impacts. Now, this multidisciplinary and transdisciplinary approach is essential to the success of our public health measures, and importantly to limiting the generational damage to opportunity across society and into the future. But global health and pandemics are not the only set of global challenges we face. Climate change likely remains our greatest existential challenge, with related consequences such as habitat and biodiversity loss, food and water security, and indeed risk of pandemics, but also geopolitical instability with involuntary migration, economic damage already evident, and so on. Now, these issues are interconnected and they absolutely require social researchers and scientists and researchers from disciplines to work together effectively to develop true holistic solutions. One of the challenges is that within universities, strong disciplines and structures can often create barriers to working across disciplinary school and faculty boundaries. So the Deakin Science and Society Network was created exactly for this purpose, to reach out across these disciplinary divides and the divides between research, policy, and practice. So the network has 4 focus areas: environmental challenges, data cultures, healthy futures, and Indigenous knowledges, and the ambition to help bridge those disciplinary divides to find new solutions, real solutions to the problems we face. It has a specific focus of supporting earlier and mid-career researchers who are addressing these challenges, looking to create science-literate social research, and conversely socially engaged science that makes an impact. And it really aspires to be an enabling platform for interdisciplinary synergies at Deakin, but certainly beyond. Now, the Science and Society Network has responded to the unprecedented challenges in 2020 with a pivot to online seminars very much such as this. And today we're following on from their very successful COVID-19 series of 7 seminars that's reached already over 7,000 viewers internationally. Now, today we have the first of 3 in the Healthy Futures theme, convened by our theme leaders, Jeff Craig and Neera Bhatia. And we're delighted today to be joined by Professor Merlin Crossley to discuss gene editing, certainly a topic that typifies very much why we need interdisciplinary perspectives on scientific innovations. Now, Merlin is both a leading Australian molecular biologist and also Deputy Vice-Chancellor academic at the University of New South Wales in Sydney. He grew up in Melbourne, completed his Bachelor of Science at the University of Melbourne, was awarded a Rhodes Scholarship and completed his doctorate in Oxford, an outstanding track record. Now, he's widely recognised for his research in gene regulation, in particular for contributions to understanding two of the most famous genetic diseases, haemophilia and sickle cell anemia. He's also been instrumental in the initial identification and cloning of key genes and coding several important new transcription factors and co-regulators. With all of this, he's also an enthusiastic teacher and, importantly, science communicator. An editorial board member of The Conversation and a key lead and advisor in a whole range of important entities within Australia. Please join me today in very much welcoming Merlin to really engage with us all in a conversation about gene editing. Thank you.
Thanks very much, Julie. Thanks, Jeff. Great to see you all and wonderful to be here. Yes, I'm in Sydney. It's Beachigal land that UNSW is on up in Sydney. And I'm just going to, after that wonderful introduction, muddle through and share my screen and begin the talk. So if I do that, technology's arrived at just the right time for us, hasn't it? So that's a picture of Sydney on a summer's winter day. And we've got Emma here doing some tweeting. So I've got my Twitter handle up there because I try to be modern with these things. So I want to be a little bit controversial today. Everyone has their own views about some of the ethical issues we'll talk about. But I really want to be provocative. And I've started with a great line from Shakespeare, friends, Romans, countrymen, lend me your ears. But I would say, I've always worried about that line because you can't really lend ears. Ears are with you for life. So I wondered whether Shakespeare, that's a misprint, and we should say, lend me your tears, because tears are something you could lend. The Romans collected them in vials, and Mark Antony could have taken some from the people who were listening. But some people are horrified at the thought of editing Shakespeare. Although I think tears is more logical than ears, people don't like to change the fundamental aspects of our culture, nor the fundamental aspects of our DNA. And it brings me to another reason why people don't like, some people are concerned about CRISPR gene editing, because they feel once it's done, you won't be able to go back. Now, many of you will know which that river is. That's the River Rubicon, which Julius Caesar crossed. And once Julius Caesar crossed that river with an army, he threw Rome into a state of civil war. I hope we never get to that point with science, but who knows? So the anxiety, I think, is related to doing things that cannot be undone. But I just begin by making the point that we do this all the time. We don't do it by introducing mutations deliberately. And the idea that we would do that to our children with germ-line gene editing, I think is highly controversial. And the immediate reaction is this, one of absolute horror, captured by Edvard Munch's wonderful picture of the screen. But what I'd point out is cultural evolution, like genetic evolution, is also in many ways irreversible, and we've been doing that for years. Teaching children new skills is what we are, particularly university people, and where many of us today are university people. We do it because it's life improving, but it also crosses generations. It's interesting, I tried to encourage my own children to learn Mandarin without success because I felt it would open up many opportunities for the future, for this century, and understandings. Many people teach their children religious views or values. It was interesting, we got a box set of the Brady Bunch, and when I watched it as a child, I didn't realize how deeply drenched in American values it is. But the values were actually I was quite comfortable with many of the values in the Brady Bunch, odd to say. And then I'm teaching my daughter to drive at the moment. And teaching someone to drive is also something that it's interesting because it reminds me of the risk that happens if you do something new. And then talking about risk again, I don't know how many of you have read this book by Cormac McCarthy. When I read this book, it took me a year to stop thinking about it. And there's one line in it: just remember the things you put into your head are there forever. But we never think about that with learning. But we do think about it when we think about genetic change. The changes we might make through germ-line gene editing would be there forever, although they could be changed back in future generations, possibly. And just to continue this theme as a provocative introduction, we have no hesitation about physical surgery or even enhancement. But I think it is worth debating, and I think we've got plenty of time to do it: genetic surgery and genetic enhancement. So if a child is born with a cleft palate, there will often be surgery to correct that condition. Neonatal heart surgery is one of the most common neonatal surgeries. And my own parents actually didn't believe in orthodontics, and they were English. But today, orthodontics is very common, an intervention for enhancement. Athletic training is another thing that's common. And I don't know if you'll notice, I don't say my R's particularly well. I took my daughter to a speech therapist and she can say her R's perfectly, but I never really learned that. So we make all these interventions which are irreversible, but we do it because we believe it improves people's lives. And here's another one. I am hugely in favor of vaccinations. I think vaccinations is a public good and a civic responsibility. But remember, when someone's vaccinated, actually the DNA and the B cells will change. So that's an example of an intervention which can be lifelong and does actually change the DNA in some of our somatic cells. So I think by this stage, you'll realize I'm quite enthusiastic about science being used for good, being used to improve things, provided we do a proper risk-benefit analysis. Now, in most of the interventions I've talked about, like teaching someone to drive, we implicitly do that. We think teaching someone to drive, especially a teenager, there's a risk there when they get their license and go off as a P-plate driver. We know there's a risk, but we think that the benefits outweigh the risk. What about with CRISPR-mediated gene editing? What are the benefits and what are the risks? Actually, I feel that both the benefits in the short term and the risks in the short term are overestimated. I think the benefits are very localized, and I'll come on to this. And I think the risks are very localized. In the long term, I think the benefits could be very great, but the risks could also be much greater. So let's talk about fixing things with CRISPR. I'm interested in fixing things to make things better, but it's not as easy as it looks with the human body. Let me explain. So first of all, what is CRISPR? It's a more efficient, quicker, cheaper way than existing approaches we have to alter genomes. We could already do it, but CRISPR is better. But let's not exaggerate how much better it is. Oh no, sorry, I meant to say this. So we've already been doing gene therapy for a while. It's difficult, but CRISPR is a better process, but it is still difficult. And this is a nice article in Science from 2016 with a nice picture and a video. One of our reporters tried to do CRISPR. He failed miserably. There's a perception that anyone can do this in their kitchen, in a laboratory, or in their garden shed. It's actually very hard to do this on human cells. So I don't think it's not one of those technologies that will be taken up widely by non-scientists. And just to explain what it is, it's a find and replace editing function. This is straight from Wikipedia. We use a guide RNA, which can base pair to find the exact place in the genome just like a editing word function, it will do a find and then the replace. It can cut the DNA to disable that word. That was the first use of it. And then the second use, if you put in a repair template, rather than just cutting and altering that word, you can get it to stitch in the new word you want. So that's a find and a replace. So originally a find and delete, but then a find and replace. And now there's things called base editors and prime editing, which are basically improve the efficiency of the find and replace function. But let's talk about fixing things. How easy is it to fix things? Could you fix a bicycle? Bikes are pretty easy to fix because you can get at all the parts from the outside, and bikes are put together one piece at a time. And so if one piece breaks, you can fix it. What about this? Could you fix a cake? If you do much cooking and if you've made cakes, if you forget to put the eggs in, or you use normal flour instead of self-raising flour, forget to put yeast in, it's pretty hard to fix a cake because things are baked in. If you forget to put the cream in the middle of the cake, you can probably fix that. If you forget to put a cherry on top, you can fix that. But it's pretty hard to fix many aspects of the cake. And what about that? Has that happened to anyone? I suspect that's happened to some people. Can you fix a phone? Phones and our electronic things today, which are disposable, there's almost been a decision that you can't fix these things. And they're made of components like a bike, but they're also sealed and things are baked in. And so I know many of you know this, but if there's anyone who hasn't had the opportunity to study biology, it's vitally important to realize that we're a bit like bikes and a bit like cakes and a bit like iPhones, perhaps most like iPhones. We're made of many, many cells, and each cell has its own DNA, like an iPhone with like a computer operating system. And the combinations of cells acting together as we grow and our bones set and our organs form is a bit like baking in the components of a cake. And we're a bit like Lego models built of lots of little phones, each with their operating system, and then baked together. Now, the fact we're like Lego, we can take bits out and put new bits in, but other parts are baked in together. So we're very hard to fix. And that's why I said at the beginning, in the short term, CRISPR will only be used for a small subset of things. It won't be used for a lot of things. So we'll have time to get used to CRISPR. And the first use will be to correct parts of our body that we can get the stem cells out of, fix them and get the stem cells back in. The ones that I work on are blood. Blood's probably the easiest tissue to get out and get back in, but it's still not that easy to get stem cells. There are heart stem cells. Getting them in and out is hard. Skin stem cells, I think, is another option. We might see some work there, or some effective work there. So if we can get cells out, modify them and get them back in, and get enough of them back in to repopulate damaged organs, then I think we can use CRISPR for fixing parts of our body, just as you would fix a flat tire on a bike. And the other thing I'd like to say, because I work on blood, I'm biased, but this is an amazing paper from 2016, which says many interesting things. But one of the things it says is how many cells we've got. And I was surprised to find out that we have many more red blood cells than anything else, by far more. Now, we don't have many muscle cells at all, but nevertheless, muscles do make up a lot of our biomass, but we've got a lot of red blood cells. And they're constantly being renewed because they get worn out as they go around our body. and you can get the bone marrow cells form the red blood cells, and you can get these bone marrow cells in and out and do work on them. They're the stem cells. But stem cells are rare. It's still not easy to get enough of them. It's not easy to modify them. It's not easy to get them back. And they have to sort of repopulate. So if the existing stem cells are still there, you won't be correcting the defects. In vivo gene therapy is not really being attempted. You could, so I'm talking about getting the stem cells out, modifying them in a test tube, and then putting them back. The other idea is just to inject things into the body. That's being done in mice, but it's not being done in humans. And then the other problem with stem cells, and this is a technical issue, homology-directed repair doesn't seem to work as well in stem cells and in other cells. And that's limited the ability of CRISPR to cause direct edits, changes. You can still use it to make deletions, but to make the desired change is much harder in stem cells, it seems, although base editing could be a way around that problem. So in summary, somatic gene therapy has been around for decades, but how many people do you know who've had it? And that's the point that I would say. I suspect very few people know anyone who's had somatic cell gene therapy, genuine gene therapy, not quackery, which is offered in some parts of the world. And I think we've got time to work out any ethical issues concerns we may have, because it will be such a small number of people opting to have these sort of therapies. So only a limited set of diseases will be treated by CRISPR-mediated gene therapies, and those are the diseases affecting tissues where we can get the stem cells out and back in, so-called ex vivo gene therapy. I think blood may be the first, anemias and various immunological diseases. Now, one day it may be possible to just inject CRISPR into someone's arm like these biohackers do and have enough of it to find its targets safely and efficiently. But at the moment, we can't do that. It's possible, it might be possible to do this into the eye or something like that, a small organ where you can really concentrate the CRISPR and do it directly into the eye. But there'll be limited examples. I work on beta hemoglobinopathies. Many of you will have learned about sickle cell disease, either at school or in university biology. It's the most abundant, most common single Mendelian disease. About 7% of the world population have a mutant gene affecting the globins, and 300,000 children are born each year with 2 mutant copies. It's because if you have 1 mutant copy of globins, you have some resistance to malaria. So you'll see it across the world wherever malaria was or is. And it's been studied. I just want to spend a moment saying this. It's been studied for more than 150 years. And we're just getting to the point that I think in my lifetime, we will be able to cure sickle cell disease. But it took so long. It was back in 1846, a researcher at London Zoo noticed that deer had sickle cells. He looked at all the different blood through a microscope. 1910, the first patient in America was observed. An African-American patient had sickle cells. Linus Pauling characterized the mutation and described this as the first molecular disease because he showed that the globin protein had a different chemical structure. It migrated differently on a gel. Vernon Ingram identified that mutation. Max Perutz solved the structure of hemoglobin. He won the Nobel Prize for that. The structure of hemoglobin hasn't helped us in understanding therapies for sickle cell, but it's been fundamental to our understanding of so much biology. Fred Sanger's sequencing technique told us exactly what the mutation is. And just recently, people have looked back and deduced that sickle cell probably arose 7,000 years ago from a single mutation in Africa, because in the heterozygote state, it conferred a level of resistance to malaria. It doesn't make people immune to malaria, but it's a milder illness because the malaria parasite finds it more difficult to live in blood, which is heterozygote for the sickle cell allele. And so people said, well, it's a problem with blood. Let's just do a transfusion. That will help. And it does help. But ultimately, iron builds up and there are problems with repeated transfusions, although you can collate some of that iron away. Then people said, well, let's fix it once and for all with a bone marrow transplant from a matched donor. And that is a good therapy. There are risks with bone marrow transplant. You have to get rid of the old bone marrow, put the new bone marrow in, and then there can be immunological effects. But it does work, but there are limited donors, and it's a very invasive procedure. Then people said, let's put a replacement gene in. That was early gene therapy done with viral vectors, but there was a lot of epigenetic silencing. So you had to lose more and more of the viral vectors. And eventually one of these viral DNAs, sometimes there was a risk it would insert next to an oncogene and cause cancer. That didn't happen with sickle cell trials, but it had happened with SCIDS trials for immune deficiencies. And now people are saying, well, why don't we just correct the mutation? So that's the obvious thing to do. That's the mutation on the slide now, a valine to a glutamate, an A to a T. But it's hard to do it because, as I said, in stem cells, blood stem cells, homologous recombination doesn't work well. The base editors can't make an A to a T change very well. But people are working on that and it can make other changes. So people are seeing whether that will work. But it still involves taking bone marrow out, modifying it ex vivo, conditioning the patient to remove the existing bone marrow and putting the stem cells back in. There is another thing you can do, and this is what my lab works on. Back again, more than 150 years ago, Janet Watson, inspiring pediatrician, noticed children had fewer symptoms of sickle cell, and it was noticed that the more sickle cell you have, the more fetal globin you have. Fetal globin's the globin you have in utero, which allows you to snatch the oxygen from the mother's blood. The more you have, the fewer the symptoms. And in 1975, there was a patient described who had fetal globin expressed throughout life. And Francis Collins, who did the Human Genome Project and still runs the NIH in Washington, he worked on mutations, the -114 mutation, which also keeps the fetal globin gene on for life. And then people found some agents, 5-azacitidine, hydroxyurea, and butyrate, which somehow keep the fetal globin gene on. Usually it goes off and the adult globin gene comes on, but if you keep it on, it doesn't do any harm and it relieves the problem with sickle cell disease. So we said, and others, why don't we just introduce the 198 mutation and put the fetal globin gene on? And indeed, that's what we're working on at the moment. We've discovered there's 2 repressors. One repressor binds here, one repressor binds there. If you can go in and edit those by, there's many ways of editing them, just the old-fashioned CRISPR way, disrupting them, or base editing, then you can turn the fetal globin gene on for life. And that may be easier than correcting the sickle cell mutation. So base editing is, I think, what I'm very enthusiastic for sickle cell disease treatment. It's good at disrupting repressor binding sites. There are very few deletions or rearrangements with base editing. So in terms of safety, I think it could be very safe. But again, the problem is you have to get the blood stem cells out and then back in. That's the real risk. And the question arises, if getting blood stem cells out and back in is a risk, would you do this again and again for every generation if this disease was in a family? Or would you say to the family, would you opt to have that disease cured once and for all by germ-line gene therapy? So now I'm going to be provocative again and talk about germ-line gene therapy. The other thing about germ-line gene therapy is that you could also use it to correct some of those disorders which I talked about being baked in. there are many hereditary diseases that affect the brain. And I think the brain is obviously a part, you can't really take a bit of the brain out, fix it and put it back in. I don't think you could do that. It's possible cells would home and find the right region, but there's a lot of baking in the brain, I expect. But with germline gene therapy, you just start with one cell. And if you inject the CRISPR into that, perhaps you could correct the whole new child and all the descendants of that child if you get it right. But if you get it wrong, you'd be causing a problem that would go on through generations. So that is a very serious thing. And at the moment, the risk of potentially harmful off-target modifications that may harm the embryo and persist for generations means that this is not regarded as ethical. And I went to this conference. I won't celebrate the name of the researcher, but many of you will know who it was. This was in Hong Kong a couple of years ago. And this researcher attempted germline disruption of the HIV receptor to render the children immune to HIV. The aim was to mimic a naturally occurring mutation. It didn't quite work exactly, but there was some effect in one of the children. It was widely condemned as unethical, risky, unnecessary, ineffective, and there were questions about the consent process and ethical oversight. So there were questions about whether it worked, whether it should ever have been done. And the general consensus was that it was premature and unnecessary. And this is interesting. Fedor Ernov is quite a famous, he was one of the pioneers of zinc finger-directed nucleases. And he says this interesting thing here on Twitter. Embryo editing, which he means germline editing, is a solution in search of a problem. There is no unmet medical need and it must not proceed. And then he talks of it, condemns a Russian scientist who wants to do the same as the previous researcher I mentioned. So if you look at that, you may think germline therapy may never be acceptable. But other people would say, if there is a medical need, then, and if it's safe, then things change. And the point I'd like to make, though, is that it would still be very rare. And the reason it would be very rare, Fedor says that there is no unmet medical need. I think he's right in that case. And generally, there won't be a medical need because there's other ways of doing things. Most families with a genetic disease, you don't need to know too much genetics here, but you will have heard of recessive and dominant disorders. If it's a recessive disorder, 3 out of 4 of the embryos will be unaffected. If it's dominant, 1 out of 2 would normally be unaffected. So you can, if you do in vitro fertilization, then you can use pre-implantation genetic testing just to identify the unaffected embryos, and there is no need to do any modification at all. The only time, and it would be so rare, you would resort to gene editing is if both parents were affected, or if there were so few embryos that you had to correct because you just didn't find an unaffected embryo. I tried to think of examples where it might occur. If 2 sickle cell disease patients met and wanted to have an unaffected child, then the only way would be through germline gene editing. But that's pretty rare. That's not going to turn our world upside down if 1 or 2 such people opted for this. One which is more higher in prevalence would be Alzheimer's risk alleles. There's a particular variant which gives a higher risk of Alzheimer's, and it could well be that in families that had a high risk of Alzheimer's, some people might say we would like to avoid that risk, and the only way would be through germline gene editing. The question is whether that would be a strong enough argument. Then people talk about genetic enhancement. This is talked about a lot. The point I'd make there is I think that will be rare as well, for a very simple reason. It's harder than it sounds. People I always like to say people are like cities. It sounds easy to make someone better, but people are too complicated. Think of a city like Paris, or think of your own beautiful city of Geelong by the coast in Victoria. If I gave you $1 million and said you can just do one thing to make Geelong better, what would you do? You'd struggle to decide, actually. And if I said you could change one gene in your own genome or your child's body, which gene would you change? You'd struggle to decide. It's very hard to know which gene to change, and you can only change one at a time at the moment. You may be able to change more than one, but it will never be that many. If you really want to enhance the next generation, it's much better to use education, training, cosmetics, orthodontics, vaccinations than CRISPR. Because the combinatorial, chaotic, and unpredictable impacts mean that the traits people really care about, beauty, temperament, intelligence, we wouldn't know where to start, which genes to start with. I do think it's possible. Someone may say, look, we can now safely disable the CCR5 gene, and that might be a way to reduce the risk of HIV infection. I think that's a possibility. I'm not in favor of that, but I think that that could be possible. I think it might be possible to say we don't want this ApoE4 risk allele. Now, there may be benefits of the CCR5 gene and the ApoE4 risk allele, so we'd want to be very cautious about that. But if someone, an individual family said, we don't want that, I think one might decide, some people might say, well, that's your decision. But here's one for you, for the long-term future. What about extra copies? And I'm nearly at the end. I'm timing it okay, I think. Extra copies of tumor suppressor genes. This is one that was put to me that I'm still pondering upon. There's a gene called P53, and it basically protects the cells from overgrowing, from replicating. And when you're young, you want your cells to replicate, but when you've grown up, if your cells replicate out of control, that forms a tumor, a cancer. And P53 stops that happening, and most cancers occur when both copies, we all have two copies of most genes, when both copies have been inactivated by just a random mutation, that's when the cancer starts growing. Elephants live a long time and they're much bigger, and people have said they don't seem to get cancer much. Why is that? They apparently have 20 copies of their P53 gene. So it's unlikely they have so many spare copies that they don't get these cancer. Many of you will be in families that have experienced cancer. Some may be in families that have a predisposition to cancer. If you could say to your children, you never need to worry about cancer because we're going to give you some extra copies of the P53 gene, would you do that? Now, this has actually been tried in mice and it wasn't great. It's not a great thing to have extra copies of P53 genes. So you need to have silent copies of the gene that only came on once the normal gene was inactivated. I think that's possible. I think that could be done. Should it be done? It won't be done in my lifetime. But I think that's an ethical question worth talking about. And then my last slide, or except for acknowledgements, to end on. I think CRISPR gene therapy, somatic gene therapy will be rare, and germ-line gene therapy will be even rarer in our lifetimes. And I think it will be opt-in. I don't think there'll be strong societal pressure for people to do it. I think some people will do it. It will be expensive. It's sort of like a heart transplant. It will only affect some families. It will be limited, expensive. I don't think it's going to turn society upside down. I think there are risks. I think the risks will be reduced as the technology improves. And those risks are for individuals. And I think to some extent, they could be decided by individuals. And I don't think the risks for society are that great from CRISPR gene therapy. I do think the risks for society are quite great from, in contrast, genome sequencing, genetic privacy, breaches and prejudice that may occur as everyone's genetic information becomes known. And then the final thing I'd say, CRISPR technologies also allow something called gene drives, which could be used to change species and eradicate things like mosquitoes and rats. That's a chain reaction. And there, I think it could turn the world upside down. So I think that's an ethical issue for us too. And then just to acknowledge, yes, I feel very privileged. UNSW's looked after me well. The ARC has, and the NHMRC, and our core facilities. That's a picture of my lab from a few years ago where we were all a few years younger, and I have gone 1 minute over time. So I will stop sharing my screen and hand back to our moderators. Thank you.
Many thanks, Merlin. A great talk. Put quite simply, I think. I'd now like to invite the discussant responses from Associate Professors Eben Kirksey and Neera Bhatia to discuss and add their perspectives on the topics. So over to you. Eben first, I think.
So thank you, Merlin, for this talk. And I'm going to agree with you on a couple of things and then disagree and try to open it up for discussion. So I very much agree that the benefits of gene editing at this point are basically kind of meh, you know? Take the gene at the center of the experiment that you mentioned in Hong Kong, done by He-Jian Kui. CCR5 is a gene that you can, maybe take out of a child and have someone grow and not experience the disease of HIV. But, there's a lot of other ways of mitigating disease risk of HIV. You can use condoms. If you're at high risk, you can take a prophylactic drug called PrEP. You can talk to your kids about safe sex. It's a lot easier to do that than knock out a gene. And, there's a lot of risks too to taking out a gene. We know a little bit about that receptor. We know that there's certain strains of flu, for example, that you might be more likely to die from if you don't have CCR5. That didn't seem like a big deal 2 years ago, but now that we're living in a pandemic, like maybe you want to keep your receptors. I also agree that this is going to be rare. And I agree in part because I've met some of these genetically modified people, these people who've gone through the first generations of gene therapy. And I know that it's very, very expensive. And this is maybe leading into where I disagree that the key ethical question that we need to be talking about here is benefits versus risks. For me, it's a question of science and justice. So the fact that it is going to be so rare, certain cancer therapies are revolutionizing the ways that these diseases are treated. I entered the Cancer Survivor Hall of Fame at Penn Medicine, where some of the very first people who received an effective gene therapy, Kymriah, the one that's the first approved by the FDA. So in this Cancer Survivor Hall of Fame, it was all white people. And this was for a disease that disproportionately impacts African-American communities. So how might we think about the ethics of equity, where it's not just trickle-down innovation, where it's not just, we're going to do pure science here and kind of let society, figure out how it's going to be used, but how do we build questions of science and justice into the very design of research agendas and programs? So another point where I might disagree just on a metaphor, you said that fixing things might be like fixing a flat tire in some cases of gene editing. For me, that metaphor falls a little flat, not to be too fast and loose with puns here, but you talked about people as cities, which I think is a much better metaphor. And we also need to think about what's broken and who gets to define what's broken. Not only do I not like that metaphor because it implies gene editing is a little too easy and cheap. Like you fix a tire with a little patch that you get for a couple of bucks, you do it in a few minutes. These are invasive, time-consuming, resource-intensive projects. But who gets to define what's broken? The disabled community has a long history of disagreement with the research community about what a normative and normal body should look like for that community. And, you know, there's been a lot of interest in sort of consulting with disabled folks about CRISPR. A lot of times they're like, okay, I'll talk to you about this, but what I really want to talk to somebody about is how to get an effective repair person to come to my house and fix my wheelchair, because my wheelchair is really difficult, complicated machine. Yeah, maybe this is an interesting technology, but I'd rather you invest your research dollars and priorities on this other pursuit. That brings me back to Dr. He-Jung Cui. So I've just finished a book. It's coming out I'll spruik it a little bit. It's called The Mutant Project. And I managed to talk to key people in Dr. He-Jung Cui's lab, the parents that signed up for it. One of the remarkable things that I'll end on is that the campus where he did this research doesn't have potable water. So the priority was this high-powered techno fix for a problem that, you know, there's a lot of other ways of dealing with, but it's this bright, shiny new techno fix versus infrastructure that we know how to create by other means. So I think I'll leave it there. Thanks.
So Merlin, would you like to address any of Evan's comments and questions?
So thanks, Jeff. Thanks, Evan. Look, those are great comments, and I don't really disagree with things. I would say with the other things can deal with HIV, it's a little bit like other things can deal with, other processes can deal with obesity. But you know what? The only thing that really works is surgery for obesity. That actually really works. Other things don't always work. So sometimes, even though there are other things, CRISPR's at the moment, yeah, I mean, I don't want to defend what happened here during Cui's lab, but sometimes the technological fix is a better fix than the simple fix because of human behavior. With the fairness of use, I hear this a lot about, you mentioned only white people were benefiting from various high-tech therapies. I think this is true, but I think that happens broadly across society. The unfairness occurs everywhere. And I think CRISPR is a part of that, but it's not really an argument which is related to CRISPR. It's just what you're reflecting on is inequities in society, which I think are very concerning, and they should be concerning, but I don't know if they can be brought to bear on every new technological advance, but that's all right. And then the what is broken thing, I have an easy answer to that. You have to ask the person. And there is this question that, is someone who's deaf, is that a disability or not? Or is it something that people want corrected or not? I would just put it in simple terms. If the person is bothered by having a flat tire on their bike, then it's broken. If the person says, that's fine, I don't ride my bike anyway, then it's not broken. But I would give the agency to the individual. So if you let the individual have the agency, then I think it's fine. But I can tell that you're an ethicist and I'm a molecular biologist. So I'm a bit cautious about some of my statements there. Back to you.
I think, Neera, do you want to go ahead with your, I've got a lot to say, but I want to let you have a chance too.
Sure, thank you. Thanks, Merlin. That was, I really enjoyed your presentation. I found it to be a very measured and rational discussion for me, and it was really easy. I like the flow. I like the beginning, actually, in particular, your first slide where you talked about history a little bit and crossing the Rubicon, and then I found there was a few philosophical points that I found very insightful. So there's a couple of points that I'd like to make from, or observations, I guess, from your presentation that I found particularly interesting and poignant. I liked your points around benefits and risks and these implicit things that we do as a society that we have no issues or qualms with. And the one that particularly stuck out for me was orthodontics, which I actually had very recently. I'm from England. I was born in England. I moved to Australia 10 years ago, and the British don't seem to pay a lot of attention to teeth. And I moved to Australia, and it was not an issue for me. It was just like, yeah, my teeth are crooked, I'm going to get them fixed. And then you move to something such as editing of genes, and it's a completely different kettle of fish. But then we think about benefits and risks of things which are essentially about improving life, or improving or extending life, or improving the quality of life in certain circumstances, when we're talking about eliminating or identifying a disease, particularly for children, some of our most vulnerable individuals and citizens, we may take a completely different approach. So I have a law background, so when as a lawyer we're looking at judgments, particularly around withdrawing treatment from critically ill infants, in these cases go to court where parents and doctors have a disagreement about whether treatment should be withdrawn, and they reach an impasse. A lot of concepts that come up regularly before the courts are issues around what are the burdens of treatment and what are the benefits of treatment? Is treatment futile? What sort of quality of life would a child have here? And those things resonate with me when we're talking about CRISPR, and what sort of benefit would this provide to a child and to its parents, and what's in the best interest. So those things really resonate with me, as if this can improve the quality of a child's life and the parent's life and society, I see that as being something that's a good thing to do. So that was one thing that I thought was interesting. I'd like to get your thoughts on whether you think it would be more palatable for society, or for certain members of society, if we talk about editing and CRISPR in developing crops over using it in terms of in humans, and whether one is more palatable to people over another. So that's, I guess, one question I'd like to pose to you. Another is when we talk about CRISPR and genome editing, people generally, I guess, the layperson thinks about it through this sensationalized lens in society of, oh, we're talking about designer babies, and we're talking about these things that are often very polarizing in society, and I put a lot of that down to the way the media portrays it. However, I'd like to get your thoughts on CRISPR and COVID as it stands, and how that may be used in terms of the pandemic. I'd be interested in your thoughts on that. And finally, my last question to you before I allow other people to speak is, from your work with, we talked in your slides, you talked about sickle cell anemia and haemophilia. And again, growing up in England, a lot of my friends were from the African Caribbean Society, and I myself have a South Asian and Indian background. And those are two diseases that are quite common in those backgrounds, ethnicities. Have you come across any of your research where CRISPR or genome editing is something that those cultural backgrounds have been more interested in, or more likely to be interested in, because they are more often impacted or affected by those diseases? Probably a lot of questions I've thrown at you in one sort of session, but I'd be interested on your thoughts.
So it's lovely to have so many questions. My parents, so to start with orthodontics, my parents were English, and so that's why they didn't care about getting teeth straightened. And if you look at English actors and US actors, in the US, actors almost always have straight teeth. In the UK, actors are chosen often for their acting ability. And you could say, I mean, I love UK crime dramas because the acting ability and the script is everything. Whereas you could be mean and say in the US, the looks are different. So there can be profound impacts of whether you go for correcting teeth or not. So I think it's very interesting, but I'm surprised that we never talk about it really, because you may as well straighten them. I sort of wish I'd had my teeth straightened. My own kids, we got them straightened because it was the thing. But it is interesting that it's so accepted, but not in the UK. Crops and agricultural animals, I think that's the answer. I think a lot of the anxiety relates to people not being sure things are safe. I mean, it's very interesting. Even with his work, there was a lot of concern, the poor babies, what's happened. When we reproduce by normal means, I don't know the exact estimate, but I think it's around 40 to 100 new mutations just happen by just normal errors in DNA replication. So there are mutations. We don't know the level there are if you throw CRISPR in as well, but it seems to be pretty low. It might be in a similar region. I think if we do a lot of work with crops and agricultural animals, we will know the error rates. And if we have, 50 years of doing that, people will become confident that it really works. It's hard to do it with individual clinical trials because we won't have the numbers, because as I think we all agree, it'll be rare. You mentioned about cultural backgrounds. I am not an expert on this, but the anecdotes I've heard are that sometimes society is anxious about a therapy, but families that are afflicted are very enthusiastic for, please do something to help us. Don't be high and mighty on your, this could be a slippery slope. Please do something to help us. Please do not. So, and I wouldn't say that for whole cultures. I don't know, but I have heard that anecdotally. And then COVID, look, the main CRISPR use there is a use that no one's, that I didn't mention at all, in detection. They were very good because it's this find and replace function. You can use the find function as essentially with some ingenious tricks from Doudna's lab and Feng Zhang's lab you can use it for very rapid detection. And I think very rapid detection, we've seen in Australia, the detection is what contains the epidemic. Isolation contains it first and then detection. So I think it's been very useful. So I think those are the points that I wrote down. So back to you guys.
Yeah, thank you. Eben, did you?
Sure, I'll jump back in. Maybe just circling back to some of those questions about kind of research trajectories and science and justice. And apologize, there's a train going on in the background, so you'll hear, we're on Zoom, everything happens. So I guess, for me, the question from the disabled community is, why aren't you investing more in wheelchair research? Why focus on this bright and shiny new tool? And I think, the broader question about, are we going to live in a society with kind of trickle down innovation where we just kind of let market forces, let kind of science shape particular agendas and live with ongoing, do we accept now that, cancer research at its baseline, social and medical inequity is just there. How do we prioritize investments right now? Kind of whose dreams matter, whose research trajectories get funded? I think that's kind of some of the questions I was trying to get at. So in a sense, we're all really excited about CRISPR. Like it seems to be this breakthrough technology that can do something. In part, your talk has kind of deflated that. So maybe a question back to you, like, should we be investing more in water infrastructure? Should we be investing more in wheelchairs? Or do you think kind of the research dollars are equitably distributed?
I think you've hit on an absolutely fascinating question, and you're spot on about it. So even in the sickle cell field, there is an argument that all of the research is about gene therapy, triumphant cures for a handful of patients. And if the same amount of money had been invested in drug development to prevent the sickle globin, they basically aggregate, sticks together and messes up the red cell. If the same amount of money had been on drug development has been put into drug development in other diseases like cancer, then it would have been cured. And then you can also say it with Indigenous health and all sorts of things, that if we invested in public health, preventative medicine, and it's the same with COVID, if we all went for preventative medicine, it would be much more effective and better value for money. But you can't get people excited about preventative medicine, and you can't get people excited about wheelchair research, and you can't get people excited about the drugs for globin, whereas people get excited about the frontier technologies. So I get worked up about this with big science. I'm not a fan of big science projects, but part of me says they're not efficient, they involve waste, they overpromise, they involve hype. But part of me says, but they're the only way. So I sort of just concede. I'm with you that there should be more research on wheelchairs. There should be more research on the basics of public health and preventative medicine. But somehow, if we try to do that, we sort of tried that and it didn't work. So getting people excited about these moonshot programs is another way of doing it. And then you've got to say, okay, let's just not spend all the money on the moonshot programs. And I'm with you there, because I think too much is spent on things like gene therapy trials and stem cell trials prematurely. So I think your point is absolutely right. But I would just argue, if you go back to your point and say, okay, we're going to cut funding for CRISPR and we're going to allocate funding for wheelchair research, just try to think through what might actually happen. It may not deliver, but I think your point's a very good one.
If I just come back in, I think I agree somewhat with both of you. And I think if we think about wheelchair research, or I'm just using that as an example here from you, Evan, and I'm just, I guess here I'm speculating, and that is that there's, I guess, research in certain things such as wheelchair research or different things that only certain members of society is going to be of interest to those members of society. And I take your point too, Merlin. If we look at sort of big projects or big technology, and I often use this word, the whole sentence around hype and hope, and people do get caught up in the hype of things because they do hope that it will lead to a cure for a particular disease that may have riddled their families. But and the other thing is, is this thing that might have once been a possibility leading to more of a probability. So I think with any of these things, we need to look at them with some caution. And I think it would be detrimental or ill-advised if we thought, well, this thing's really hyped, so we should just discard it because it's just a possibility. It's a possibility rather than a probability. I think we need to still look at things such as gene editing, because that is, I think, something that is likely to happen in the future. Although I think we need to be cautious as to how much money is invested into these projects. But to just say, it's hype and it's never going to happen, I think it would be wrong to think that way.
Yeah, I mean, you have to harness the hype for hope in a way. It's sort of interesting. The hype drives me nuts as well. And I think most serious researchers are more annoyed at hype than they are wedded to the hype. But on the other hand, inspiration and some of this, you know, perhaps it is a force for good in the long term. It's interesting. There's an interesting comment just come out in the chat from Chris Mays at Deakin, talking about how commercial biotech, there are impacts on millions of farmers in the global south. And yeah, I want to acknowledge that point, that it's not a neutral testing ground. And yeah, I guess the reason I overlooked that point is because, yeah, I'm so focused on my own research in the health sector and humanity that I don't think of that. So that's a good comment in the chat there. And back to you guys.
I was wondering if we might circle back to He Jiankhui's story a little bit more, and less his perspective, but the perspective of the people who signed up for the trial. So I had the privilege of talking to some of the parents and understanding that for them, so HIV in China right now is much like it is in Australia or the US. Your life expectancy is very similar to normal. There's predictable and actually even free access to antiretrovirals there. But at the same time, there's profound social stigma. And one of the motivating things for these families that signed up for the experiment is an unmet social need. So, you know, their health was basically taken care of by this policy that's about 15 years old now, guaranteeing everyone medicine for life. But they wanted their children to never experience this profound social discrimination. The irony is that, you know, in the intensity of the moment, the children were called monsters by mainstream media, and more intense stigma than you know, the average person who has HIV. But the other reason that they signed up for the trial is for most participants, it was the only legal way that they could have a baby in China. So if you're HIV positive, you're prevented from using, sperm washing is one technique that you can use. You go to a clinic, you give sperm, it runs through a centrifuge, the viral particles get separated out from the semen. And, you know, there's some people who have enough money to travel abroad, make babies that way. There's others who work out various, quasi-legal ways to have children. But the hopes of these parents were tied up in a child, and this experiment was the only way to do that. So maybe it's a roundabout argument for suggesting that not only do we think about unmet medical needs as we evaluate the ethics of a new technology, but that we also consider unmet social needs, and how to address those in the specific and in the broader senses.
It is interesting to learn that, Eben, because He Jiankhui seemed to be very sincere that he was doing it and providing a service to the parents who wanted it. And I wasn't sure about whether they wanted it, because they were afraid of HIV, because of the stigma, because there wasn't good treatments, or as you said, because there wasn't another way of having a child. And it was clear that he felt that he was meeting a need that was there. He didn't feel like he was tricking people. I think people didn't necessarily accept what he said at face value at the time, but now that you've got further information, it's interesting. It complicates it. And then the question is, if people want things for themselves, if they have negative effects on other people or society, you can say no. But if they don't have negative effects for other people in society, on what grounds do you say no? other than it's a waste of money, which I think you've touched on, that it would be better. There are much cheaper things and better things, and prioritizing is one of the most ethical, challenging things in life. How do you prioritize, going back to your potable water thing, in this society, how do you prioritize all those things? But that's great that you've done that research and got to the story, because I think it certainly influences my thinking on it.
And I might also say, yeah, so it's such a complicated case, and there are so many things, and I think your quote from Fyodor Urnov is very spot on in terms of how he was thinking about it. One of my chapters is called "A Hammer Looking for a Nail." He was initially looking at a gene associated with cholesterol before a Communist Party official convinced him to switch to HIV. And super complicated case. I think the only scandal in the participant consent process was the presence of the participant consent process. I had the privilege of watching a video of it. And the big scandal for me is that Michael Dean was in the room, his PhD advisor from Rice University. He Jiankui has been charged with seeking fame and fortune, with practicing medicine without a license, but his US counterparts have not been charged. Kind of switching tacks a little bit, going back to some of the earlier discussions about decision-making and sort of individual autonomy in decision-making. I think one of the things to appreciate as we're moving forward with technologies like these is how society and the sort of norms that we hold, but also the institutions that frame and shape the healthcare decisions we make are all impinging upon the kind of choices we might make if CRISPR were ever to enter the clinic as a germ-line technology, for example. So again, some of my disabled friends are worried that they're going to be edited out of existence, not just the deaf community, but again, what counts as broken and what counts as normal. In some of the databases, XXY is regarded as anomalous. So Klinefelter syndrome, which produces an androgynous body, someone who's not going to be, gay or bisexual. It has nothing to do with sexual orientation, but it produces a body that's gendered differently. And there's liable to be many other things, many other kinds of choices that one can make about one's offspring. So I think it's about thinking about these distributed societal norms, but then also, our insurance company is going to be dictating the kinds of choices that a prospective parent might make. It is if and when this technology is perfected to the degree that it's predictable, it's affordable, is there going to be an imperative to make babies not the old-fashioned way, but with CRISPR and to sort of correct a suite of genes. So these are some of the medium to far future things I think that are worth thinking about.
So I think those are valid questions, but to keep them in perspective, they won't be with us in the short term. They'll be with us in the very long term. I mean, I think in the short term, it will be life-threatening diseases in somatic cell therapy. I don't know if we'll see any germ-line in our own lifetimes. And I do think, though, if you look way into the future, it could be the case that there would be some societal pressure or health insurance derived pressure for corrections. But, that pressure hasn't even affected the UK to adopt orthodontics yet, so it's going to take a while. I don't want to trivialize it because orthodontics is trivial in comparison, but some of the things you're saying are complex things where things aren't really broken. I think that what will happen is a natural priority that the major things will be done first. I think Fyodor Urnov is excessive in his condemnation of it. But on the other hand, I think he's right. There just isn't going to be an urgency at the moment. So why do it? Why do it? So I think we've got plenty of time in the very, very long term. So I heard this quote, and I don't know who it's from, the first stage of civilization is developing language and speaking. The second age is learning how to write so that we have history and we learn through cultural evolution. The third stage is learning to write the genetic code. It is possible in the very long term that we've unleashed a genie that will change everything, and there could be control and influences. But I think we've got time, and we've got guys like you and Nia and people to debate these issues. I think we've got time.
Okay, thanks. I have a couple more questions. One is for me, relating to what you and Eben were just talking about. I heard a quote from a deaf parents once, that they'd rather their child be deaf than not deaf, so they could help them best, they could communicate them best. So in the future, as you said, what if a deaf parent said they wanted to actually edit, if they wanted to edit their child or their prospective child to be deaf rather than not? And I guess that's a particular scenario, but it could apply to other conditions as well. And it relates to the way that we view what disability is, both in society and both to a family, with something that we call a disability, but may work differently within a family.
That's amazing, Jeff. That's the trolley problem. That's the trolley problem, in that you're happy for the trolley to go and hit the one person rather than the five, but you won't push the one person over the track because that's actually using agency. So look, that's an amazing one. Here's a simpler one. What would you say if the family says, we don't believe in vaccination, so we're not getting our child vaccinated, and then the child grows up and then sues its parents for not being vaccinated, or sues its parents for having the deaf gene. So this is an interesting one. The one thing I've learned is that the topics related to children, vaccinations, and even the teaching of language, phonics, goes crazy on social media. People feel very strongly if you interfere in a parent's ability to bring up their own child. So I'm going to let the others talk about that for the moment.
I got one more question of the audience. This is a question from Namitha Mohandas, who's a PhD student of mine, just graduated, in fact. Her question is, isn't it important to invest in basic science research rather than gene editing, considering we don't really understand how genes interact in a systemic scale? How can we guarantee that gene editing, even for a particular disease, would be safe and would not lead to risks in the future?
So that's a great answer. And there is an example, I can't remember the gene that, there are two different variants, and one gives you a predisposition to one disease and one to another. And so it's a trade-off. And these genetic interactions, like in a city, will be infinite. What we're doing with the globin genes is only introducing naturally occurring mutations that we know of. He Jiankui also tried with CCR5 to introduce a naturally occurring mutation, which was known to be safe in a Caucasian population. And people said, well, how do you know it would be safe in non-Caucasian populations? I think some of these gene interactions are pretty minor, because genes have evolved moving around and they tend to be compatible. So some of them are minor, but some of them could be profoundly important. So it's a good question. And I think it's a safety question. It's saying, be careful what you change, because you don't know what the unforeseen consequences are. And I see that in science, but I'll tell you where I see it a lot more in management. You try university management, everything you do has negative unforeseen
No comments.
effects. So yeah, it can happen, but it's less so in the genome.
A related question goes back to what you were saying about turning the fetal globin gene on for life. Now, you mentioned that there was the odd individual with that gene turned on, but in a wider population, how much can you really determine from, say, mouse studies, and how much would be kind of trial and error? What does it do in late childhood, middle age, old age? How does it interact with cancer? How does it interact with blood disorders, infection, etc.? How do you proceed with doing that in the human population?
So there are sufficient patients with hereditary persistence of hemoglobin that I think there is enough public data.
Okay.
But in some cases, there won't be enough public data. But what I would say is that, how do we know that this seminar today is not going to give people nightmares? I mean, we can never be absolutely sure that things are safe. And sometimes you have to make a judgment call, and you make a judgment call by putting the known benefits over the potential risks, and there could be risks. But if you find out that the therapy is doing harm, then you stop it. And that's what happens with some drugs, where people have found, oh, this is a good psychotic drug. No, it's not. It causes this side effect or that side effect. So sometimes I think you give something a try, but you correct your action if you're found to be mistaken.
I think some of the detractors will mention drugs like thalidomide, but then thalidomide has come back recently, being used for other conditions as well. So in some ways, it could be reckless to say this, but nothing ventured, nothing gained. And it talks about people's changes to real people. But I think what's important is that people, the audience and others, are given, listen to the arguments for and against and the various nuanced parts of the discussion, so that will enable them to come up with their own decisions as well. So I think this discussion and similar ones will stimulate people, both scientists and lay people, to actually think a bit broader than their narrow area. As you said earlier on, you didn't think about this particular topic, and you acknowledged how it was good to have feedback from ethicists and from lawyers. And personally, I've benefited from discussions with ethicists, lawyers, and sociologists. And I think that's where these discussions should go. We should have a thorough discussion so people can make up their own mind about how they would like research to proceed. Any other further comments from Eben or Neera?
Yeah, I just want to, on the back of your comment there, Jeff, I think that's a really valuable point that you make. I think this is a topic that cannot be looked at in a silo. I think it's something that requires a wholesale evaluation from lots of different disciplines, because it's something that will impact society and lots of different parties, and therefore it does require a social, ethical, legal, scientific evaluation, and I think that's really, really important. Going back to the point that we touched on a little bit earlier with Merlin and Eben around children, I can't remember who made the point around children and how parents have and social media. I think that was the point, and I think that's a really, really important point. I've looked at this in a different context, but children bring out the best and the worst in people, I find, when it comes to a discussion that involves children and parental rights, and there's a lot of unfortunately misconceptions around what rights parents should have and what rights parents do have, and at least from a legal perspective. And I think it will be very interesting, and I personally am of the view that I don't think for a very long time, I think I might be in, I don't know if this was where Merlin was going, but I think I'm of the view that I don't think we're going to be anywhere near that stage in the short term, at least, where we'll be having these discussions, or at least where they'll be requiring any type of court intervention as to whether parents are having to agree or disagree to any type of treatment for children around this. But it does bring out the worst and the best in parents, and there's this misconception that parents ultimately have the right to demand treatment for children, and it ultimately, in most cases, the courts become the ward of the child, and there's parens patriae jurisdiction, whether the parent of the child. So we think of parents as being the ultimate protector and person that has the dutiful rights over the child, but the child doesn't belong to the parents. So it will be very interesting to see what happens in these cases in the future, and how regulation and regulatory frameworks are built around this space. I think, to be honest, we're far too early in this whole space is far too premature and very much in its infancy to see where this goes. But I think yeah, there'll be a lot of discussion by parents and even other people that feel that they're making the right decision for children. And as I say, it brings out the best and the worst in people at the best of times.
Evan, did you have one final point?
Yeah, I have a parting shot on metaphors. And for starters, just wanted to say, Merlin, I was really delighted at all the diverse metaphors that you used, and wanted to offer one other one as we're kind of thinking about issues of risk and safety. In explaining CRISPR, I often use a drone. So, with the drone, you can give it GPS coordinates. It can go somewhere in the genome, and it can blow things up. And sometimes it takes out the target, but sometimes it takes out the wedding party. And then sometimes it goes astray and gets the GPS coordinates slightly wrong and blows up the wrong thing. One thing on risk, in talking to some of my friends who signed up for what was billed as the first editing experiment. This is the Sangamo zinc finger experiment that Fyodor Urnov and colleagues were working on. They're walking around with billions of genetically modified cells in their body. And each time that drone strike happened in one of those cells, the dice was spun and a different pattern of genetic damage emerged. 2 out of 4 of my friends who were genetically modified in that experiment have had prostate cancer. The jury's out as to whether just older men get prostate cancer or if this is from the experiment. And in trying to get answers to basic safety questions, the first decade or so of gene therapy, there was about 4,000 people that participated in those experiments, but very few long-term studies have been done. We work on grant cycles that are much shorter than decades or lifespans. And I think there's a real opportunity now to do some of that longitudinal stuff to try to figure out some of these basic safety questions. So a parting metaphor and a parting suggestion that we all be cautious and rein in the scientific hubris a little bit. But overall, I loved your talk, Merlin, so thank you.
Thanks, Evan. And I'd like to thank Merlin for his wonderful talk. And I'd like to thank Evan and Mira for introducing some very, very interesting and useful discussion points. A brief quote to finish with: "Since I'm a thought, the possibility potentially one of the most important concepts to arise in the history of mankind." And he concluded, "Indeed, this concept marks a turning point in the whole evolution of life. The first time in all time, a living creature understood its origin and could undertake to design its future. Even in the ancient myths, man was constrained by his essence. He could not rise above his nature to chart his destiny. Today, we can envision that chance and its dark companion of awesome choice and responsibility." So I will again thank everyone and just share this screen to advertise our next talk in this topic. And it is by Hannah Brown from Monash University on communicating with impact, telling science stories that matter. My name's Jeff Craig, and thank you all for tuning in. Thank you.